Topic is genes from cell and molecular biology. First, let's see about definition of the gene. The gene is to genetics what the atom is to chemistry. The gene is the unit of genetic information that controls a specific aspect of the phenotype. The gene is the unit of genetic information that specifies the synthesis of one polypeptide. Let's see about terms related to gene. Recon. It is the smallest unit of DNA capable of undergoing crossing over and recombination. Muton. It is the smallest unit of DNA which can undergo mutation. Cistron. It is the unit of function. It is a gene in real sense capable of synthesizing a polypeptide chain of an enzyme. Complon. It is the unit of complementation. Next, let's see about classical concept of gene. It was introduced by Sutton in 1902 and was elaborated by Morgan in 1913, Bidge in 1923, Muller in 1927 and others, which outline as follows. Genes are discrete particles which is inherited in Mendelism fashion that occupies a definite locus in the chromosome and are responsible for expression of specific phenotypic character. Number of genes in each organism is more than the number of chromosomes. Hence, several genes are located on each chromosome. The genes are arranged in a single linear order like beads on the string. Each gene occupies specific position called locus. If the position of the gene changes, then the character changes. Genes can be transmitted from parent to offsprings. Genes may exist in several alternate forms called alleles. Genes are capable of combined together or can be replicated during a cell division. Genes may undergo for sudden changes in position and composition called mutation. Genes are capable of self-duplication, producing their own exact copies. Let's see about modern definition of gene. Gene is the unit of genetic information. That is, the sequence of DNA that specifies one polypeptide, which includes coding as well as non-coding regulatory sequences. Modern concept of gene. S. Benzer in 1957 coined different terms for different nature of gene and genetic material in relation to the chromosome on the basis of genetic phenomena to which they involve. Genes as unit of transmission or criston, genes as unit of recombination or recon, genes as unit of mutation or muton. First one is genes as unit of transmission or cistron. The part of DNA specifying a single polypeptide chain is termed as cistron. A cistron can have 100 nucleotide pairs in length to 30,000 nucleotide pairs. It transmits characters from one generation to other as unit of transmission. Second one is genes as unit of recombination and recon. The smallest segment of DNA capable of being separated and exchanged with other chromosome is called recon. The recon consists of not more than 2 pairs of nucleotides. Third one, genes as unit of mutation or muton. Muton is the smallest unit of genetic material which when changed or mutated produces a phenotypic trait. Muton is delimited to a single nucleotide. Then gene types. On the basis of their behavior, the genes may be categorized into following types. First, basic genes. These are the fundamental genes that bring about expression of particular character. Lethal genes. These bring about the death their possessor. Multiple gene. When two or more pairs of independent genes act together to produce a single phenotypic trait. Cumulative gene. Some genes have additive effects on the action of other genes. These are called cumulative genes. Pleiotropic genes. The genes which produce changes in more than one character is called pleiotropic gene. Modifying gene. Gene which cannot produce a character by itself but interacts with other to produce a modified effect is called modified gene. Inhibitory gene. The gene which suppresses or inhibits the expression of another gene is called inhibitory gene. Let's see about gene action. The influence of gene resulting in the expression of a genetic character is called gene action. The genes are generally associated with the production of enzymes which they synthesize from chemical substance available in the body cells through a process of autocatalysis. As a rule, one gene affects one enzyme. The various actions of genes are expressed in their development of pigments, colors, hormones, size and form, production of proteins, antigen and antibody, production decisive effect on human diseases like albinism, thyrocyanosis, etc. Genes in protein synthesis. Jacob and Monod proposed operon model to explain the mechanism of gene action. Protein synthesis is regulated by three specific genes located on chromosome, such as structural genes, operator genes, regulator genes. Structural genes. It regulates to produce specific mRNA. It determines the kind of protein to be synthesized. Operator genes. These genes act as switches to turn on or turn off the activities of structural genes, regulating the elongation and termination of polypeptide chain. Regulator genes. These genes produce certain proteinaceous substances called repressors, which prevent the operator genes from their action. Let's see about the essential features of genes. It determines the physical as well as the physiological characters. It is situated in the chromosome, occupies a specific position known as locus. These are arranged in single linear order, occur in the functional states called alleles. Some have more than two alleles known as multiple alleles. Some may undergo sudden change in expression called mutant gene, which is called as mutation. May be transferred to its homologous or non-homologous counterpart. Can duplicate themselves very accurately, which is called replication. Synthesize a particular protein, determines the sequence of amino acid in the polypeptide chain, which is termed as the genetic code.